Thanks, organizer invited me to give a talk. So actually, my lab is more interested on cancer metabolism, more focus on cancer biology, but also this conference giving me opportunity to learn. That's my dream to looking for metabolomics connect with kind of signaling pathway. So actually, they connect with the previous two speakers. So in cancer biology, about 20 years, we most focus on oncogene signaling pathway, and also pharmaceutically, also looking for directed target oncogene to try to treatment cancer. But unfortunately, we know in cancer treatment, a lot of drug is failed for direct target oncogene. There's a lot of reason heterogeneity tumor, and also can tumor have redundant pathway. So and look now is difficult only targeting oncogene, or at least only targeting one oncogene. So is difficult for kind of treatment. So recently, about 10 years, cancer metabolism is getting into cancer biology field. The reason because scientists found oncogene signaling pathway can directly regulate metabolic pathway. So like PI3K pathway, a lot of another pathway can regulate metabolic enzyme. That's why the two field connection now. And more interestingly, metabolites can come back reversely regulate the oncogene signaling pathway. Like acetylation, that can come from glucose acetyl-CoA, can acetylate a lot of protein. Most famous is the histone protein. And also like methylation, epigenetics, all is coming from metabolites. So that's why these two field connection now, and also bring us more insight to how to target cancer. So to talk about cancer metabolism, we have to mention Otto Warburg. Actually, he is getting a Nobel Prize because he found cytochrome C. But his most famous work now to be recognized is he first observed glycolysis. So he found so in normal cells, so usually glucose to be oxidized in mitochondria, fully to produce ATP. But Otto Warburg observed is in tumor tissue, even in normal cell condition. Usually in normal condition, glucose have to enter into mitochondria to produce ATP. But he found is in tumor tissue, even in high oxygen condition, glucose continually generate pyruvate to lactate. This is different with normal cells. That is his observation. This is 1930s. But since then, we don't understand why tumor is shift from oxidative glucose oxidation to glycolysis. So we don't understand. Even now, we still don't. We know the tumor cell is addictive to glycolysis, but we still don't know why, the reason. So, but currently, we mostly in cancer biology, we agree to the proliferating cells, not only tumor cells, like embryo cells, all is highly proliferated cells that require abundant nutrients to support duplicates of biomass. This is very simple to understand because we know the tumor, we know the cell divide, they need biomass. So then where they can get biomass, they can get them from nutrients. So in proliferating cells, particularly in tumor cells, we know the tumor cell can uptake a lot of glucose. What does glucose go to? Not only go to ATP. Actually, glucose can convert to protein, nucleotides, lipids. And so this is, a few years ago, also scientists find that tumor cell also can uptake a lot of glutamine to convert to macromolecules to support biomass. That is how can cell to get these nutrients to support biomass duplication. So that's why currently in pharmaceutically, targeted cancer is looking for metabolic inhibitor. So this previous speakers already mentioned how can inhibit the metabolics, so to inhibit tumor growth. This is pretty hard topic now. Like we probably know IDH1, IDH2 mutation inhibitor, PKM2 inhibitor. So all is developing now. So, but my field is coming from glioblastoma. This is brain tumor is grade 4 astrocytoma because when I work at postdoc at UCLA, my boss is working on glioblastoma. So, but we work on traditionally signaling pathway, have EGFR, PI3 kinase, AKT signaling activated glioblastoma. And from there, I shift from the signaling pathway to metabolism. So in GBM is very famous. Is you have all upstream target kinase receptor EGFR is amplification or mutation in around 45% of patients is activated, and accompanied with other this called is HER2, PDA receptor A, and MET, all is application, and together with NF1 deletion and P10 deletion, so together we activate the PI3 kinase AKT pathway in around 88% of patients. So that is very promising to targeting PI3 kinase AKT pathway in GBM. So that's why beginning to, since 2005, so targeting EGFR and EGFR3 use olatinib. So it's pretty interesting in glioblastoma. But unfortunately, the drug is failed. The drug don't have any effect in GBM. The reason is have a lot of reasons, but one of the reasons is because the drug can cause other target kinase receptor reactivation. This is either from beginning small population already have this mutation, small population this mutation, application of mutation, or reactivate this pathway, but we don't know exactly the mechanism. So, but my interest is we avoid targeting upstream because all upstream have mediated by either by RAS-ERK pathway or PI3 kinase AKT pathway. So my hypothesis is whether we can find the common target to avoid direct target upstream to kill tumor cells. So come back cancer metabolism. So the cancer metabolism now is most this is only a little bit of metabolism. We focus on now is glucose glutamine to biosynthesis pathway. So in cancer metabolism, actually PI3 kinase AKT pathway can directly upregulate the glycolysis mediated by this glycolysis enzyme to promote the glucose enter into mitochondria or go to lactate. So they have a lot of different ways to regulate lactate and mitochondria, PDK1, PDH. This all kind of cell through HIF they can mediate this pathway. I don't mention that. So the question is, glucose actually can enter into TC cycle, then secrete citrate into cytoplasmic. Then cytoplasmic can synthesize as precursor to synthesize lipids. So this is how the most of the glucose, a lot of glucose can enter into the lipids in not only the normal cell as well in cancer cell, a lot of cell. And also actually now we know is glucose and also can enter into nucleotide synthesis through PBB pathway and through serine glycine pathway. So this is how glucose to convert to macromolecules. So my interest is we know is PI3 kinase pathway can activate glucose in tumor cells. PI3 kinase can enhance glucose to lipids. So I have hypothesis during my postdoc at UCLA. I think is PI3 kinase AKT pathway have to directly activate lipid synthesis gene. Then to let these flags from glucose to lipids flags to smoothly. Otherwise, if only upregulate the glucose, not upregulate lipid synthesis gene, the pathway, the flags, the metabolic flags cannot smoothly go to the lipids. So then I converted to, but this is not a direct jump to here. So I spent a lot of half a year. I figure out SRBP is fit in this pathway. So not like simple. So SRBP is a sterol regulatory binding proteins. This is the major transcription factor to regulate fatty acid synthesis. They have 3 isoforms. SRB1C is regulator, S1C and 1A regulate fatty acid synthesis pathway, and SRBP2 is regulate cholesterol synthesis. And also, actually, we demonstrated is in cancer cells, SRB1C or 1A directly regulate LDL receptor. This is Braun and Goldenstein, they get a Nobel Prize. They describe this scheme. But this thing is SRB2 mostly regulate LDL receptor. But our data show is in cancer cell is SRBP1 is regulate LDL receptor. So how SRBP2 be regulated? So the SRBP is when the translation, this is no function, only this protein localized in ER membrane. Then they can bind in SCAP and INSIG. Actually, INSIG is a negative regulator. They can sterols can bind in INSIG, then INSIG can bind into SCAP, SCAP binding SCAP, SRBP. They can hold this complex in ER membrane, so they don't move. Then when sterol level decrease, then INSIG will dissociate with SCAP. Then SCAP is SRBP activating protein. Then SCAP will translocate SRBP to Golgi. This through COP2 vesicle. This is vesicle trafficking. So then after enter into Golgi, then they have 2 enzymes, S1P and S2P, will cleavage SRBP sequentially. Then only N-terminal will enter into nuclear. Then the nuclear form SRBP can transcribe lipid gene expression. This is how to SRBP to regulated by sterol. This is described by Goldenstein and Braun. So, but the interesting is in cancer cells is different. What the difference is because in cancer, even sterol level is high, SRBP is still very activated. The difference is Braun and Goldenstein described. So the interesting we found is EGFR PI3K AKT pathway can upregulate SRBP, but the mechanism still don't know yet. So that's my laboratory now focus on how EGFR pathway can regulate in which step to activate SRBP. So we can see this is glioblastoma patient sample. We can say SRBP is a nuclear form, really highly upregulated in patient tissue. And also, we found in vitro cell line, when activate EGFR, V3 can increase fatty acid levels, demonstrate EGFR V3 correlate with fatty acid level in cell line. Then we have this GBM patient autopsy sample. We compare tumor tissue with normal tissue. So we found that SRBP is highly, in tumor tissue, highly, N-terminus highly upregulated compared with normal tissue. And also this upregulation correlated with EGFR phosphorylation and correlated phospho-AKT phospho-6. So demonstrate SRBP1 activation is correlated with EGFR PI3K AKT pathway. Then we quantify 140 patient GBM patient sample. We found in normal tissue all is negative, very low level. And in patient tissue, SRBP1 level and downstream ACG factor this is a lipid synthesis gene, all correlated with EGFR, EGFR3 phospho-AKT levels. So the activation. Then further to demonstrate the EGFR mediated by PI3K AKT pathway promote SRB1 activation, because P10, the P10 is a negative regulator PI3K AKT pathway. If P10 is intact, this pathway should be decreased. So we have patient sample we show is phospho-EGFR high, but P10 is very low. Then phospho-AKT increase, SRB1 is activated. And in patient sample is even phospho-EGFR is high, P10 is high expressed, and phospho-AKT is decreased. Then SRB1 is also low. So the demonstrate is EGFR pathway in patient is regular SRB1 mediated by PI3K AKT pathway. Then we have in vitro cell line, we re-express EGFR-V3 cell gene in UD7 cells. We can see activated phospho-AKT and also increase SRB1 cleavage. Then when we re-express P10 level, so we inhibit the phospho-AKT and also downregulate SRB1. So demonstrate this pathway makes sense. So then we have lopatinib clinical trial sample. We have patients. This lopatinib can target EGFR and target HER2. But in GBM, usually HER2 is very low level. So we have this clinical trial designed by pre-treatment. We get a biopsy sample. Then after 10 days treatment, we get a second biopsy sample. So the reason is designed the experiment because there's a lot of drug after 3 months, 2 months treatment, all population is different. So you cannot examine pathway. That's why this pathway we can show is for EGFR pre-treatment is high, SRP1 high, and after the treatment, all is go down. Demonstrate this in clinical sample is SRP1 is correlated with EGFR activity. Then we summarize. Actually, we also measure the PTF receptor c-MET signaling pathway through PI3K AKT mediated SRP1. All is similar result. Then we have hypothesis if we inhibit SRP1 directly, whether we will break this connection and kill tumor cells, particularly in this kinase activated tumor cells. Then we use SRP1 lentivirus, knock down SRP1, we can see can dramatically inhibit separation, particularly in V3 cell line. Then also we can see V3 cell line actually induce cell death. So demonstrate V3 cells actually more dependent on SRP1. Then pharmaceutically, we use a natural 25 hydrocholesterol. This can directly bind into in-seq, block SRP1 activity. We can see this drug can inhibit the SRP1 cleavage and decrease downstream acid fast gene expression as well. Now we can see in this drug treatment, we can see actually V3 is more sensitive than UD7. So this so the VEGF is quantitative activity EGFR forms. And then we screening different cell line have higher phospho-EGFR, they have higher SRP1 levels, but lower EGFR will have lower SRP1. Then we test whether the drugs have specifically targeting SRP1. So we can see in lower SRP1 cell line actually is less response to the SRP1 inhibitor. Then in higher SRP1 cell line actually can kill tumor cells. So demonstrate targeting SRP1 is very sensitive for tumor cells with SRP1 activation. Then come back is cancer metabolism. Then we know is EGFR promote SRP1 to increase fatty acid synthesis. Then we targeting SRP1 can inhibit tumor growth. Then we ask the question whether we can target fatty acid synthesis is downstream of SRP1, whether we can phenocopy SRP1 effect. So we treat with this tumor xenograft model. Then we treat is C75 and also plus atorvastatin to block fatty acid synthesis and also cholesterol synthesis. We can see can dramatically inhibit tumor growth. So demonstrate this kind targeting lipid metabolism is one promising pathway to inhibit the tumor growth. And also very interesting show is this is tonal staining. We show is green one is endothelial cell. This demonstrates the targeting lipid is can induce tumor cell apoptosis. And very interestingly, actually I just come back from cancer metabolism meeting, keystone cancer metabolism meeting is endothelial cell in tumor cells because of high proliferation in tumor cells. Our drug also can directly inhibit blood vessel endothelial cell apoptosis. It makes sense because of endothelial cell is highly proliferated in tumor tissue. So to summarize, so actually in normal tissue or in lower EGFR signaling tumors, actually the pathway EGFR mediated mTOR can promote the protein synthesis and also mediated by SRP1 promote the lipid membrane biogenesis is very low. That's why tumor grow very slow. Then in high EGFR signaling, like high grade tumor, they have both pathway very high. That's why tumor will grow very fast. So how we can target the tumor? So now we have some disagreement because people think is mTOR can directly regulate SRP1, but our data show is mTOR actually partially regulate SRP1, mostly regulated by AKT. So we show is when we're targeting SRP1 downstream pathways, and this will cause tumor dramatically kill tumor cells because the tumors continually produce protein, but not produce fatty acid and not produce membrane. That's why actually to imbalance tumor cells can kill most dramatically kill tumor cells. So and also now our lab is very interesting to looking for SRP1 inhibitor try to can translate to clinical trial. So and also to summarize what's going on in our lab now, so actually we show is glucose actually they can go to TC cycle and use a precursor for fatty acid synthesis. And also actually we have another paper show is in tumor tissue is tumor can uptake LDL mediated by LDL receptor. We demonstrate the LDL receptor is highly upregulated in tumor tissue. And we found is, but SRB2 is regulated de novo synthesis cholesterol actually is not really high in tumor tissue because it can get the cholesterol from blood and uptake. So, and also glutamine can enter into TCA cycle, also can convert to fatty acids. So then we, the SRB1 is the role is regulate fatty synthesis gene and also directly upregulate AD receptor. So that's connected with oncogene. So oncogene can actually activate glycolysis pathway and also directly regulate SRBP1 is mediate several mechanisms, but it's not very clear. That is a major pathway we're looking for now, and also actually also another paper show is how we increase cholesterol efflux to reduce cholesterol level to inhibit tumor growth. So that's okay. We have several directions to going on now. Is we work on autophagy, how we involve this lipophagy, how to involve the lipid metabolism to contribute to tumor growth, and also how signaling pathway regulate SRB1 and radiation, how radiation affect this pathway. And actually, we're looking for hypoxia effect in tumor tissue, a lot of hypoxia condition, and also we're looking for microRNA targeting lipid metabolic gene to treat the tumor. So actually, this is just collaborators at the OSU. Thank you.
Are they viable?
So we didn't not directly knock down SRB1, but we data show is even we use drug SRB1 inhibitor in SRB1 lower expressed tumor cell actually is a very less response.
Yeah, but it doesn't. Yeah. What you use doesn't just do that. It hits.
Yeah, in normal, that definitely we need to test that.
And the other question is the structure of SRBP1 such that it's a druggable target?
SRB1 is because SRB1 is transcription factor directly target transcription factor is difficult, but it's now is actually can target the upstream to block SRB1 cleavage pathway. Yeah.
Other questions?
The major challenge with targeting the upstream targets is most of the inhibitors targeting the EGFR, the patients they become resistant to it over time. So how can it be tackled?
You mean protect EGFR?
Yeah, the most of the drugs like erlotinib and lapatinib, the patients get resistant over the time.
Right. Actually, in glioblastoma, we cannot predict all the patients resistant, no effect. So the reason is we know the EGFR inhibitor erlotinib actually works in lung cancer, very good response, but even is get resistant. But in GBM is no patient is get benefit. So that's why now it's harder to predict because every patient actually erlotinib is not going on in GBM. Even now with the combination erlotinib with Avastin or the other drug. But the reason how now we just need to understand why tumor is so resistant to erlotinib EGFR inhibitor, because I think is now one of the reasons is because tumor is heterogeneity. In GBM is EGFR tumor only have 45% application. They have other population is not.
Wouldn't it be more logical to use dual inhibitors, like targeting the upstream target as well as the downstream one, like the dual inhibitor which targets EGFR and AKT as well?
Yeah, actually this already developed in a lot of kinds of type, targeting EGFR, targeting PI3K, ET, targeting mTOR. Actually they have a drug dual target PI3K and mTOR. So actually the ongoing clinical trial, but unavoidably all the tumor will develop resistance for this one.